Hi, my name is Kim Leonberger, and I'm an Extension Associate with the University of Kentucky in the Department of Plant Pathology. And today we're going to be talking about plant diseases. By this point, you should have already completed the video regarding plant diseases and the introduction to these different organisms. So the goal of this video is to provide you with some samples and to allow you the opportunity to kind of meet and greet some of these different fungi and bacteria and viruses that you have been introduced to in the original recording. So as we go through today, we're going to talk about each one of the samples. You will be provided with many of these samples in person, so you can take a much closer look at them, but we'll talk about the views that you're going to have of them and what you would be looking for, and a little bit about each one of these diseases. So before we get started and start talking specifically about these different samples, I want to introduce you to your favorite tool, which is called a hand lens. And that's this device right here. So this opens up. It's also sometimes known as a jeweler's loupe. So you open it up like this. The ones that we have that we like the best are a 20 times magnification. And they also have a little light right here, which is an LED. So this can be really helpful when you're looking at samples. It gives you a little extra light, sometimes makes it a little easier to be able to see what you're looking at or looking for. So let's talk about how you actually use this tool. So in many cases, the way that people think of using a hand lens is like using a magnifying glass where you would kind of hold it out away from you. But in fact, that's not true. It works much more like a pair of glasses. So you're going to keep this up close to your face. So you use whichever eye you prefer. I use my right, so I hold this up to my right eye. I close my left. And then you're going to use your other hand to be able to bring that sample close enough that it comes into focus. And generally, this is pretty close. It's usually not all the way out here. It's usually much, much closer to your face. That's going to give you a really good view. And this is going to give you 20 times magnification over what you can generally see with just the naked eye. So this is a really handy tool to be able to have. These are available from a lot of different retailers, including many online retailers. If you need specific suggestions on where to find them, please talk to your county agent. They'd be able to direct you. If they're not sure, they can definitely get in touch with me, and I'm happy to direct you to some different sources for these. Now, hand lenses can come in many different forms. This is the version we like the best, but many others might come in something that looks like this. So you can tell it looks a little bit different here. And in this one, we actually have two hand lenses or two jeweler's loupes. So we have a smaller one that is a 20 times magnification and a larger one that's 10 times magnification. And you would use these the same way. These do not have lights, but sometimes, a little less magnification can be helpful. So it kind of comes down to personal preference, but some level of magnification is generally helpful. As you learned in the introductory material, most of these pathogens are much too small to see with the naked eye, although sometimes there are clues and indicators that these pathogens are present. And that's what we're going to talk about today as we look through some of these samples. So in order to do that, I'd like to get started and talk through these different samples. So we're going to start first with powdery mildew. This is sample 1 in your kit. So this is powdery mildew on a peony plant. And what you can see here when you just look at it and make some general observations is we've got this white powdery growth, which is very characteristic and where powdery mildew gets its name from. So this particular pathogen is going to have this really white growth. That is actually a fungal sign. That is the fungus itself. Now, when we zoom in a little bit closer, we can get a better look at that fungus. So here we're starting to see some different kind of more characteristic white powdery growth that is, again, that characteristic symptom and sign of this particular pathogen. So powdery mildew likes to overwinter in plant debris. It likes to be on soil surfaces. So all of these leaves that would fall off the plant are going to continue to hold on to that pathogen. So it's really important to clean those up at the end of the season. So again, what we're noticing here is that we've got this white growth. Powdery mildew is one that generally when we look at it, it's pretty easy to kind of see and tell that it's powdery mildew. Powdery mildew can be on lots of different types of plants. Peony is a common host for this particular one. But then Things like zinnias, some of our lilacs, certainly a lot of our cucurbits, so things like our cucumber and squash and zucchini can be susceptible to a powdery mildew fungus as well. So this is a fungal disease that will get in and cause this infection. Usually likes those kind of warm spring temperatures, and it starts to work its way up the plant from the base usually, or from the inside out. So once we have looked at it from this standpoint, next, we're going to want to really zoom in, and this is where those hand lenses really start to come in, because what we can do is we can take a closer look with those hand lenses, and that's what I've done here, is try to capture with a camera using a hand lens what you might see when you're using hand lenses and looking at these samples. And what you see when you do look at this is that right here, we have a really nice little fungal colony. So again, this is a sign. This is that mycelium that we talked about in the introductory material. That's those fungal strands. So this is the body of a fungus. And so that is a sign that we're actually seeing. And so again, with powdery mildew, what will happen is throughout the season, every 7 to 14 days, we will see where it will go through a reproductive cycle. And within this colony right here, you will have where it is producing more spores or offspring that are going to go and cause additional infections. So again, one of the management strategies that we talk about with powdery mildew specifically is making sure that we get that plant material cleaned up at the throughout the season. So if you start to see this disease come in, you may want to take it out so that it can't complete that life cycle and go on to cause additional infections. So removing those infected leaves early on, or as soon as they're detected, can be very, very beneficial. So one of the things that powdery mildew does like to do is that it actually can be really prolific. And you can see here where it can actually start to really take over leaf surfaces. And this is where we start to see more of the damage coming in, where we start to see that there is it's blocking that plant's ability to complete photosynthesis. So this is where we start to see more of the damage. Typically, powdery mildew is not a death sentence. It just makes plants look ugly for a while. But when we do have severe infections like this, where it's really taking over leaf surfaces, we might start to see some early leaf drop or a lack of leaves, just because that plant is being really impacted. And that's where powdery mildew can really cause a lot of damage. Typically, we don't see that. This is kind of an extreme example. Now, one of the other things when we take a close look with our hand lens that we see are these black peppery flecks. So it kind of looks like pepper got spilt on these leaves. And the importance of these structures is that this is how this pathogen chooses to survive the winter. These are the structures it uses. And these overwintering structures are small balls, and they actually have little hooks on the edges of them all the way around. And the reason why that's important is because it uses that structure to be able to cling to things. It clings to plant material. It will cling to any decorations or stakes or tools. It can also be on the soil surface. So this is why that concept of sanitation that we talked about so extensively in the introductory program is really important, because what we're doing when we do that sanitation piece is removing all of this potential inoculum from the field. So that can be very, very important to really mitigating and limiting the amount of disease pressure that you might see in subsequent years. So typically, one of the kind of misnomers out there that happens with powdery mildew is that it's on the top, only on the upper leaf surface, but that in fact is not true. Powdery mildew can be on the top of the leaf, or it can be on the bottom. Sometimes it can be on stems or produce as well. So in this particular case, this is another example of powdery mildew. This is sample number 2 in your kit. And in this case, we're looking at oak leaves. These are leaves that have already fallen off the tree. And again, you can see that white powdery growth that's happening right over here. And this is on the lower leaf surface. So this would be the side that would be facing the ground on these leaves. And what we see as we look at this is with our hand lens is that we actually, again, we have those black peppery flecks. Those are again, overwintering structures. Sometimes you can see these without the aid of magnification, but certainly having that magnification can give you a much closer look. And again, cleaning these up is really important because understand that each one of these individual little black flecks can hold somewhere in the neighborhood of 15 to 30 spores. That's a lot of inoculum just in this small area of the leaf. So you can imagine if you've got hundreds of leaves that are infected and have all of these overwintering structures, how much potential inoculum is really sitting out there. One of the things, just a reminder, if you are going to clean up that plant debris at the end of the season, is to make sure that you do not compost it. Make sure that you burn it, bury it, or destroy it. Destroy it meaning take it off site, or put it into some sort of trash receptacle, or some larger communities may have commercial pickup of yard waste, and those do go to commercial compost piles, which do get hot enough to kill these pathogens. So again, just making sure that you're looking for these structures. When we see these overwintering structures, or again, we see that white mycelial growth, that is where we actually know that we have a fungal problem. And specifically with this one, because that white powdery growth is so characteristic of powdery mildew, it really clues us in that what we're looking at is that fungal pathogen of powdery mildew. Next, we're going to switch gears a little bit, but we're also going to talk about kind of more of a leaf spot, which tends to be a really common question that we get, is that there are spots on some sort of leaf material. And in this particular case, we're going to look at maple, and this is a disease that's called phyllosticta leaf spot. And in this particular one, what we have when we just look at the sample, this is sample number 3 in your kit. This one, you can see that we are starting to see these really characteristic lesions starting to form. They start out as these small little spots, and then over time they can expand and they can get quite large. So this again is a fungal disease, and we're going to zoom in a little bit, and we're going to take this as, we're just maybe looking a little closer with our eyes next. And so here we're looking specifically at that lower left-hand side of that previous picture where we have several lesions that are forming. And again, what we notice when we look at leaf spots, it's really important to be specific about what we're seeing. So we see some brown centers, we see some darker edges. They do look like they're expanding. Some of these even have kind of a reddish color around them that maybe indicates that this isn't the only spot where the pathogen is, that it's actually kind of moving outward. And so then this is where we would again get out our hand lens and take a closer look. And so that's what we're seeing here. We're zoomed in on this one specific lesion. And when we do that, what we can see is that we actually right here in the center have that little black dot. And what that is that's what we call a fruiting structure. So this is where all of those spores are being produced. Oftentimes there will be multiples of these within a single lesion. So you can imagine the amount of reproduction that's happening in each of these lesions if there's going to be multiple fruiting structures. So this fruiting structure that we're seeing, again, is a sign. That is the actual evidence that there is a fungal pathogen present in this sample. So again, knowing that that's what you're looking at can really help you discern that, yes, there is a disease here. And then at that point, that's where you can seek out further help if you need it, or if you're very familiar with this disease because you've seen it lots of times, that gives you kind of the confirmation that you're looking for. Again, this is one of those diseases that overwinters in plant debris. So again, that sanitation piece just is going to keep coming up because it is so important when it comes to doing this. This is one that we oftentimes don't have a lot of other management recommendations for because, again, it's not going to kill the tree. It just makes it look a little bit ugly, sometimes leads to some early defoliation in some cases when disease might get severe. But fungicides with this particular one are not going to be a common recommendation unless it's a really high value tree or it's being applied by a certified arborist with equipment that can get really good coverage. Again, when we talked about fungicides, we talked about how important it is to get really good coverage because making sure that that pathogen is coming into contact with that fungicide is what really allows that fungicide to work. And you can imagine how difficult that might be if we're talking about a very large maple tree with lots of leaves and a very dense canopy, being able to get that fungicide in there to come into contact with that pathogen in all the places that it's existing. can be really difficult. So again, really the sanitation piece here is how we manage this disease. And so again, just causes some kind of yucky looking leaves for a while. And again, just cleaning those up, burning, burying, or destroying, as we previously talked about. So now we're going to talk about leaf spots again. While sometimes we do have that obvious fruiting structure when we use a hand lens, sometimes that's not always the case. So here what we're looking at is a disease that's called strawberry leaf spot. This is sample number 4 in your kit. This particular leaf disease is a fungal leaf disease. However, as we zoom in, what you're going to notice is we don't see the fruiting structures like we saw previously. So you can notice on this particular one, this is a very common disease of strawberries. We see this frequently. We start to see these leaf spots forming. We can look a little closer, just looking closely at it with our eyes. And this is what we see. We've got these really washed out centers surrounded by these really dark rings. And that's when that fungus has gotten in and caused disease. And this is looking at it under with that hand lens. So really that 20 times magnification, being able to look at this. And again, what we see is something that doesn't really give us any indication of what we're looking at. We're not seeing those obvious fruiting structures. We're not seeing any mycelial threads. So this is one where this could be a challenge when we talk about diagnosis, because we're not going to see a sign. So it's important to really use those hand lenses to look and see what you're seeing or what you're not seeing. Are you seeing fungal threads? Are you seeing fungal reproductive structures? If so, that really leads you down that path that yes, we've got a fungus present in this sample. But just because we don't see that doesn't mean that it's not fungal. And so it's really important to make sure that we take the time to investigate and collect as much information to walk through that diagnostic process, because certainly in a situation like this, where we're not going to see any obvious signs, the diagnosis of this sample could be really tricky, especially if you're not familiar with this particular type of disease because you've never seen it before, which is pretty common. All right. So next we're going to talk about a disease that is very common. We see this a lot. This is called cedar apple rust. And this particular disease is a little bit more complex than some of the other ones. It is a fungal disease. And what you're looking at here are some leaves from an apple tree. And what you'll notice with this is that we've got these really characteristic yellow spots. They start out really small, and then over time, they get larger. So we can see right here is probably something that would have been how it started. So just these little yellow dots. And then over time, they start to get a more, I guess we'll call orange or red appearance. You can see a lot of these kind of have like a red in the center. Or we're starting to see some dieback and necrosis. So again, we're seeing some really characteristic things happening here. So again, this is an apple sample. Cedar apple rust is one of those diseases that can be on apple or crab apple. But this particular disease can be a little bit more complex. But let's take a closer look before we start talking about why it's so complex. So when we zoom in and we use our hand lens, this is what we see. We can see that within each one of these spots, there are actually these fruiting structures. And this is where reproduction is happening. So this is where this fungus is going in and it's creating more spores that are going to go and are going to actually create more disease within apple and crabapple. Now, cedar apple rust is a complicated one, as I mentioned, because it actually has 2 hosts and requires 2 hosts as a way to be able to complete its life cycle. Now, the particular fungus that causes this disease can produce up to 5 different types of spores. So the structures that we see here indicate that there is a fungal pathogen present, and these particular ones are going to continue to cause infection and disease within apple and crabapple. However, at a particular point in the season, what we're actually going to see is that out the bottom of these lesions, so on the lower side of the leaves, we actually have some protrusions that come out, and those produce a different type of spore. So this particular disease can produce up to 5 different types of spores that all have various functions. So that spore that they produce at the end of the season is actually going to go and impact its alternate host. which is cedar and junipers. And so at that point, they go to that plant and they cause a very different looking symptom. They actually cause a gall, which is what you see right here, this very large tumor-like looking structure. Now, these galls take about 2 years to mature. And once they do, they start to put out these kind of orange jelly-like, we'll call them tentacles, out of this structure. And that's where they're going to go and release spores that are then going to go back and impact your apple and your crabapple. So keeping these two different groups of plants far apart is important. Now, obviously, due to the Kentucky landscape, there's probably not a place in Kentucky that I know of or have ever seen where we're not within at least a half mile of a wild cedar or juniper. And we know that these spores can travel up to a half mile. So it's important to keep them as far apart as possible. And while that doesn't completely eliminate disease, it can reduce it because these organisms are very small and those spores have to work really hard to travel those long distances. So while they can do it, it definitely is more difficult for them. So the greater space that you can have between them, the better. There are resistant varieties of both. So if you do like both and you want them close together, you certainly can do that, but we would highly recommend that you look for some sort of resistant variety of both apple or crabapple and cedar or juniper, whichever plants you're selecting there. So let's take a little closer look at this particular gall because under some magnification, it starts to look very different and very cool. This is looking much closer with just the naked eye. So this is taking just kind of holding it closer to your face, and you can start to see where those kind of orange tentacles are starting to come out. So we harvested this one before it got all the way to maturity and all the way to putting out those tentacles. And this is using your hand lens. And so you can see a lot more closely what those different structures look like. So again, these long structures here that are coming out, this is where you're going to see the production of those spores that are going to go and impact your apple and crabapple. So again, there are fungicides available for this particular disease when it comes to apple and crabapple. Typically with cedar and juniper, if you see the galls forming, as soon as you see them, cutting them out can be really important because again, they do take up to 2 years to mature. So if you see them coming on and they're quite small, making sure that you go ahead and cut those out before they have the chance to mature really cuts back on your disease pressure as well. And the leaves for this particular disease on the apple and crabapple are sample number 5 in your kit. And the actual gall is sample 6. So let's switch gears a little bit and let's talk about some woody tissues. We've talked about leaf spots and we've talked about damage on leaves, but let's talk specifically about woody samples now. And I want to talk about cankers. So cankers are essentially the lesions like we see on the leaves, but those are lesions that form on woody tissues. And they can really have a variety of appearances. They can be something like what we see here, where we start to see this cracking that's forming. You can see this. It can be this flat tissue. You can see that this right here just isn't rounded. See, it's very flat in this space. It can be where bark is literally falling off. All of those indicate the presence of some sort of canker. Now, cankers are really damaging and oftentimes go really unnoticed until they get severe. And the reason is that once you have a canker, like what we're showing here, that creates a blockage. So now the plant can't move water and nutrients up that branch. And so we start to see dieback and decline in certain areas of the tree. Oftentimes, if we trace that decline and dieback back, we can find some sort of canker that indicates where the problem is. Now, the disease that we're looking at here is again, a fungal disease. Like everything that we've talked about up to this point is fungal. This one is a disease that's called chestnut blight. And we touched on this a little bit in the introductory presentation as well. Now, chestnut blight has a very long history. And so in this particular case, you can really see the damage that it's causing and how it's getting in and causing that canker to form right here where this comes in. However, it's important to note that cankers don't always look very obvious. There, I think if you saw that cracking, you would probably be very, you probably really, know that there was something going on. But what about something like this, where you have a much smaller cracking surface and maybe it's facing up in the tree, so it's not as obvious? It can really be a challenge to find these sometimes. So you take a really keen eye. And certainly when we're talking about larger trees that might get cankers, this can be really difficult, because oftentimes we can't reach where these cankers are, or we can't reach where, can't get to where we need to be able to even look to see if they're there. So one of the things that is really important with cankers is a lot of times we can be really effective from a management perspective if we just prune them out. So just cutting out these infected branches. Now, the important thing to note is that when you find the canker, so when you find that that space where you've got the really obvious cracking, like down here, this is not the edge of the disease. The disease has moved further up into the branch. So when we're making our pruning cuts, we certainly don't want to just cut on the edge of where the cracking is. We would want to look for what we call a gradient. So you can see that as you move this way down the branch, we end up in this space where we have a really interesting transition zone. So this is kind of the edge of the disease right here, where we can at least see symptomatic tissue. Now, the disease has probably already moved back further into this branch. So it would be really important when we make our pruning cuts that we really look at where we want to make that cut. So when you make your pruning cuts, we really like it if you go 8 to 10, preferably 12 even, inches behind the last visible symptom. And by behind, I mean towards the base of the tree. So, really tracing that back, giving yourself a lot of space, making sure that you're making that pruning cut cleanly, so that the tree can heal itself properly, but making sure that you get out all of that disease. If you're really close to a branch union, you can go ahead and make that cut right at that branch union as well. You can go ahead and take out those last few inches if you're going to be really close to that union. When we make pruning cuts, the best time to do that is typically in the winter, once the tree has gone dormant. We really like the January, February time scale. We know that trees are dormant at that point. Typically, we're not going to get, some crazy weather that's going to kind of force them into opening up. So we really like that January, February time. You can prune throughout the growing season, but the important thing is that you want to make sure that you never ever prune right before it rains or right after it rains. Give yourself a 24 to preferably 24 hour window on either side. So if you're going to have kind of a 3-day stretch where you're not going to have any rain, making sure that you go out and do your pruning cuts, then making sure that if you are pruning out disease, clean those pruning tools as frequently as you can. Making sure that you clean it after every cut is ideal. There are lots of different products that you can use to clean tools. Certainly the one that seems to be best from the research that's out there is bleach. However, bleach can be corrosive on tools, so making sure that you rinse that thoroughly can be really important to making sure that you preserve the integrity of your tools. If you are going to use bleach, 45 seconds in a 10% bleach solution for 45 seconds. So dipping it in there or soaking in there for 45 seconds, that's all you need. And then making sure that you go to clean water to rinse that, and then making sure your tool is completely dry before you make your next pruning cut. A lot of times we'll get questions about if people are pruning, if they should paint anything over that wound. We don't recommend that. We typically say let the tree heal itself. They do a pretty good job of that. So we tend to like to let them just kind of run their natural course. Sometimes in painting something on there, we can possibly even trap moisture or some sort of pathogens that might just be floating around against it and really create a petri dish that kind of then leads to a bigger issue. So pruning cuts can be really important from that standpoint. Now, when we look at these samples under some level of magnification, we can actually see, I'm going to go back one slide there. We can actually see that, again, we have little bumps, and that's something, if you could feel the sample, you would feel the little bumps. And those are those fruiting structures. Again, that's where reproduction is happening. That's where they're making more spores that are going to go and cause additional infections. This is where the pathogen likes to hang out over the winter to be able to just be waiting for, some sort of wound or insect damage or some sort of way to be able to get into that plant tissue. While you don't have a sample of chestnut blight, you do have a sample of Nectaria canker, and this is sample number 7 in your kit. Nectaria canker is a common fungal disease. Again, we do get that canker, so we can see that there is cracking that's happening. Oftentimes, this one will produce a more subtle canker where we're just seeing a really flat spot, so it should be rounded, but you'll just see where it kind of just looks flat on one side like this. And that indicates that you've got a canker in that space. One of the interesting things about Nectaria canker is that it produces a really obvious fruiting structure, which are all these little orange bumps that you can see on this sample. And when we look at them under a microscope, you can see that they are just really interesting to kind of look at. But again, this is where that reproduction is happening. But with all of our canker diseases, really pruning them out is the best way to get rid of that plant material. And again, making sure that if you are pruning out diseased material, don't leave that lay at the base of the tree. Make sure that you're collecting that and that you're moving it off site or burning it to make sure that you're destroying those pathogens, making sure that they can't just work their way back and undo all the hard work that you just did in pruning them out. Next, I want to talk about volutella blight. So volutella blight is a common disease that can be on our boxwood trees or our boxwood plants. And so what we start to see is that in these shrubs, we'll start to see this kind of discoloration happen. We'll start to see, leaves kind of turn these really straw colors. We'll start to see dead spots happening in those plants. And so this is what the sample looks like. This is sample number 8 in your kit. And so when we take a closer look, just kind of zooming in, looking at the naked eye, we can start to see that there's some things going on here. One of the things that we can notice is that some of these leaves kind of look like they have something growing on them. That indicates that maybe we need to take a closer look with a hand lens. We can also look at some of these stems a little closer, some of these kind of more woody tissues, and be able to see if we're looking for any cankers there, because sometimes we can find those as well. And in fact, that's what we can see here. This is one of those stems that's kind of at the base, I guess you would call it, of the sample that we have. And what you can see here is that we have some really obvious cankers happening. We have this cracking that's happening right here. And again, so that's kind of indicating that there's something going on. That's why we're starting to see that decline happening in the base of in the upper canopy of the plant. And when we use our hand lenses to look at those leaves a little bit more closely, this is what we see. What we find is that we've got these kind of clusters of spores that are happening here. This is again where that reproduction is happening. This is that fungal sign. These indicate the presence of a fungus. In this case, this is volutella blight. It's a common stress pathogen. So boxwoods that tend to be under a large amount of stress weren't installed properly oftentimes can lead to this, or winter damage is a frequent thing that can happen to boxwoods that can then lead to this volutella blight coming in. Typically, we're going to say prune this out and get rid of that plant material. Certainly, there are other management options such as fungicides that exist, but really pruning this material out can really lead to a successful outcome. One of the things that is challenging with the management of this one is that typically we grow boxwood in very dense settings, which really facilitate this disease. And so pruning some of that out can leave some holes, and sometimes it's very unattractive. So you may have to be strategic about how you would go about pruning out that, but also making sure that before you let that plant get too dense, because certainly that's going to, again, make this disease worse. It's going to create that humidity that this disease really, really likes. So again, we know we have a fungal issue here, and that's what we're seeing with these particular samples. And again, this is volutella blight. All right, so let's kind of switch gears one more time and let's talk about roots. So this is one of those things that we tend to forget about, right? Because roots are underground, we don't see them, we don't think about them. But certainly roots are integral to the success of a plant. And when roots don't establish well, or when they become infected by pathogens, certainly what we start to see in the upper portion of the plant is that decline and dieback. And so we'll often, again, say that when you start to see something like decline or dieback, that you really need to trace that back down and really look, one, for cankers, like we talked about, but then also looking for, if you can find any shallow roots when we're talking about very large plants, or if we're talking about plants like annuals, some of your flowers, some of your vegetable plants, even being able to extract those from the ground, dig them up, take a look at the roots. If they're great, put them back in the ground. If they're not, maybe that's where you take that whole plant in as a sample to talk with your county agent. So in this case, what we're looking at is actually a root rot disease. This is a fungal pathogen, and this is a root ball from a Japanese holly. And so in this particular case, when we look at some of the roots, you can see that there are some that we really like, some nice thick roots here, like these, or like these down here. These are good roots. These are nice. They're well established. If this sample wasn't as old as it is, we would see that they would be kind of creamy. These are a little bit more woody, but typically on our annuals and some of our other plants, we typically like a nice creamy white root. Some plants have various colors, but creamy white is usually the good roots. However, when we look a little bit closer, we can start to see that we have some areas where maybe we don't have so many roots, or that we specifically have some, like this one right here, where we've got, it's really thin, really, really thin. So again, looking for those, and those indicate that what you're looking at is something that is not quite right. So what we want to do is take a little closer look. This is under the magnification with a hand lens at looking at those roots. Now, I will say that if you're looking for a fungal disease, you're not going to find obvious signs. Typically, rotted roots are going to be black. A lot of times they will slough off. So like an outer casing will kind of slide off the root. Those indicate that those are not healthy roots, that they could be infected with fungi or water mold pathogens. And so it's important that if you're seeing that and you're not able to discern, okay, it is a fungal pathogen or it's not, that's when you have that conversation with your county agent. If they're not certain, they can send that on to our diagnostic lab, one of our diagnostic labs, and be able to get some more information for you, because our diagnosticians are able to look at these under a higher level of magnification. And in many cases, what they do is they actually take those roots and actually try to get the fungus to grow out of them, so that they can identify which particular fungus might be infecting these roots. So again, just knowing that if you are seeing those roots that are really thin and frail, and are black and look really gross or mushy, those aren't healthy roots, and that could be the reason why you're seeing the decline in that upper plant canopy. And again, there's no sample for this one. Rotted roots are rotted roots, but they're not a whole lot of fun to look at. So next, we're going to move away from fungi. Everything that we talked about previously fell into that fungal category. So that big category, remember that 80% of our disease issues fall into that fungal or water mold category. And so that's why we have some more samples. Typically, we're able to see some of those signs from those different organisms. And so they can be a lot more interesting to look at, and they make much better samples to kind of introduce you to some of these pathogens. But now we're going to talk about some bacterial samples. I've got 2 bacterial samples that we'll talk about. Understand that with bacteria in this particular case, we're not going to see signs. It's very rare to see bacterial signs. You really need a microscope to be able to do that, or you would the only case where it's really obvious are those bacterial streaming or oozing, where you would see those. But again, those aren't terribly common experiences. So what we're looking at here is a disease that's called fire blight. We talked about this in the presentation, in the introductory presentation. But in this particular case, remember with bacteria, they have to get in through a wound or a natural opening. And with the fire blight bacteria, the main way that it enters plants is through the flowers. And so what we can see here is that these were once flowers and now they've started to mature into fruit. And this is where the pathogen entered. And you can see that fire blight gets its name because it's got these really big, this really dark brown, black color that makes the material look burnt. And again, blight is a term that gets used frequently, but what that means is rapid dieback. So we start to see this decline. So these bacteria came in the spring, they've moved back and further into the plant. So let's take a little bit different look at this one. One of the characteristic symptoms of fire blight is what we call this shepherd's crook. So that's this turning over of the very end of the branch, just kind of looks like it fell over. It's a very key characteristic symptom. And so when we start to see fire blight come in on apple, crabapple, ornamental pear, all of those kind of pome fruits, we we'll see this start to happen. So this happens within a single season. You can lose kind of some of these thinner branches within a single growing season. So looking for that shepherd's crook can really be an indicator that what you're seeing is fire blight, even though we're not seeing any specific signs that go along with that. When we don't manage fire blight, and again, the main way that we manage fire blight is we're going to prune that out. So marking those specific branches or limbs that need to be removed throughout the growing season, and then pruning in the winter months can be a good way to do that. Or if you'd like to remove them during the growing season, doing that, we would observe the same pruning principles that we talked about with fungi. So we're going 8 to 12 inches behind the last visible symptom towards the base of the trunk. Or to the nearest branch union, never pruning right before it rains, right after it rains, and making sure that our tools are thoroughly cleaned in between each pruning cut. If you're going to prune in winter months, it's important to make sure that you mark those branches. It's really easy to find fire blight when there are lots of green leaves on trees. It's not so easy to find them when all the leaves have fallen off and you're kind of just looking for discolored branches. So marking them in some way can be a helpful way to really look for those branches that need to come out if you're going to wait and prune in the winter months. When we don't manage fire blight, it actually can cause cankers. And so this is an example of where we've had some fire blight that's gone unmanaged or has moved back further in the plant and been allowed to kind of continue on for a period of time. And that's where we start to see these cankers forming. So we have one right here that's real obvious. You can see how we've got this dark plant material. You see it's cracking, it's malformed. You can see we've even got some concentric rings looking like it's starting to form here. But we also have another canker, and that's this one right here. Keep in mind that fire blight can go unmanaged, but over time it will kill the tree. It will go back into the base of that plant and cause a canker right at ground level. And at that point, pruning is very easy because you just have one cut to make right at ground level. But oftentimes that's not the case that we want. So making sure that we rely on these pruning principles can really effectively manage this disease. One of the other things that we say with this particular disease that's important to note, there are resistant varieties out there. So if you can look for resistant varieties, that can help you as well. So making sure that you rely on resistant varieties as well as pruning can really effectively manage this, as we have a very limited amount of compounds that could be used because we're talking about bacteria now and not fungi. So we would not use fungicides on these products or on these bacterial samples. We would need to use antibiotics, and we like to save those for human use, not so much for treating our apple trees. So again, relying on those other management strategies is really key for managing these diseases. The next disease I want to talk about is crown gall. Crown gall is a very interesting disease in the fact that while it's a bacteria, it starts to have a much more broad host range and can impact a lot of different types of plants. In addition, it also can reside in soil for a period of time. So it's important to know what can be impacted by crown gall and what might not be. Crown gall, when it gets in, it actually is a bacteria that gets into the plant and it causes the plant to produce a bunch of undifferentiated cells or this gall or tumor. And so over time, these start out kind of small, like the one that you see in the top part of the picture here. But if left unmanaged, they can actually get quite large. And of course, when these galls form on these stems or in the root systems or on the trunks of trees, they can actually girdle them. So what that means is it's going to choke off all of that plant's ability to move food or nutrients and water. And once you can't move all of the good stuff that the tree or plant needs to keep living, that is when we start to see this decline. And there's nothing that we can really do about this. So it's really important that we look for plants that are either resistant or are non-hosts and put those specifically back into spaces if we're going to take out a plant. Here, this is a sample that's on euonymus, which is a common landscape cover, but I've seen this on lots of different types of plants. It can also be on a lot of our fruits. So it can be on things like our raspberries or different brambles. There it tends to favor being in the root system. So you don't necessarily know that you have a problem until you dig up those canes in that root system, and then you start to see these galls forming. On grape, it looks a little bit different though. It actually causes those vines to really kind of break apart. They kind of look very gnarled, almost looks more like a canker as opposed to a gall. So same disease, same pathogen, causing very different symptoms on different types of plants. So again, we're not going to see any signs with these bacterial diseases, but they can cause a very interesting range of symptoms. And knowing that they're there and taking them out can really provide you with the best management. So now we're going to switch gears again. We're going to talk about viruses. As with the bacteria, signs are not going to be something that you see. You won't see signs with viruses at all. And so we really just look at them from a symptom standpoint. When it comes down to diagnosing these particular samples, it can oftentimes be very difficult, challenging. Most cases, we can narrow it down to one of two options, which is either a virus or it's some sort of chemical damage. At that point, having the plant history, having the information of if it's possible that it could have been exposed to some sort of chemical compound can really be helpful in letting us know what we're looking at. Understand that it being exposed to a chemical is potentially a better situation than if it's got a virus. Viruses cannot be cured. We can't do anything about them. And they really pose risks to a lot of different plants. We have to be really, really diligent about removing those plants that are infected so that we don't see spread to other types of plants in our landscapes or in our gardens. So making sure that we really denote history is really important when we start talking about these viruses. So what we're looking at here is a virus sample on peony. And this is sample number 11 in your sample kit. In this one, what you'll see is that we just got a really kind of range of symptoms. We've got some mottling where we are getting a yellow color mixed with a dark green color. You can see that we've got some ring spots and line patterns happening here. Let me kind of highlight where we're looking at some of those ring spots. So you can see kind of up in this area, we've got some ring spots happening here. Or even here. And then we certainly have just a really wide range of appearances here. And so this entire plant was infected with a virus. We would need to do extensive testing to be able to figure out specifically which virus it was impacted by. But again, when we start to see these symptoms, our mind goes one of two ways. It's either virus or it's some sort of chemical damage. And so again, that's where that plant history just plays such a critical role. And our last sample is number 12, and this is rose rosette virus. And this particular disease impacts all types of roses, including knockout roses, which had become very, very popular because they were known to be resistant to most of the diseases of concern for roses. However, this particular virus is a concern for those knockout roses as well as other types of roses. Once the plant is infected, again, we recommend that you dig it out because we do know that it's been shown to move through root unions where, two roots might cross from two different plants. With this one, we get a really wide range of symptoms. You can see here that we've got some red color to the leaves. We can see the thorns. We're going to zoom in on those here. You can see the amount of thorns. Certainly there can be other symptoms that go along with this particular disease, things like stunting, dieback, decline, legginess, misshapen flowers. I mean, there's just a whole range of various symptoms that can go along with these particular viral diseases. The good news about this particular disease is that it's been shown that after a period of time, you can actually put roses back into that same space, assuming that you don't have any additional disease pop up. So taking them out and getting rid of that plant material that's infected is really critical to them. Maybe if you want to be able to put roses back into that space, being able to do so. This particular disease is moved by an aerified mite, so it's a little insect-like creature that does not fly. It has to be blown on the wind, or it has to be able to crawl from plant to plant. So increasing plant spacing can certainly help, and making sure that you have proper management can be important as well. One of the things that can be really difficult with this particular disease is oftentimes roses have a very emotional connection for us. There's something that a family member has grown before and given to us, or something that is very historical. And so there can be a temptation to say, well, I could take a cutting of it and I can save that. Unfortunately, that is not the case. So please don't be tempted to do that, because essentially doing that and rooting that and then putting it back into the space then poses a risk to other valuable roses that might be in your collection or in your landscape. So making sure that, unfortunately, you take those plants out and go from there is really the best option and the best management for that. So that includes all of our samples that are included in your kit. So you should have a total of 12 samples. If you do have questions or are confused by anything, please reach out to your county agent. They're able to help you. They really like to be able to talk with you. If you have plant problems now or in the future, please reach out to them with a phone call or an email. Find out how they'd like to get that information from you. If they run into issues or need additional assistance, they are backed by a wonderful team at the University of Kentucky of specialists and diagnosticians who can help them work through that. And we're happy to get you answers and recommendations for the plant problems that you're experiencing. So I hope that you've enjoyed the opportunity to get to know some of these different pathogens and see some of these samples and the damage that they can cause. Do understand that the samples that you've looked at today are often extreme samples. They are kind of the worst of the worst. We do that intentionally because it makes it a little easier to see what you're looking at. However, most cases when we see a disease coming in, if you're scouting your plants regularly and you're able to keep an eye on them, being able to remove infected plant material when you start to see it, recognizing that there might be a problem and seeking out help and guidance at that time helps you be able to use management strategies earlier and makes them more effective. So again, if you have any questions, please don't hesitate to reach out to your county agent who has access to my information as well as our team of specialists. So thank you so much for participating in this program. I hope you have a wonderful day.